Continuing on with Chapter 5, we're going to start to look at bacterial and viral genetics and genomes. Again, here is the chapter outline for your reference. Now, some of you may roll your eyes when we start talking about bacteria. Oh, they're so boring, just little germs that get me sick and things of that nature. They actually play a big role in all sorts of genetics outside of themselves. We actually use a lot of the machinery from bacteria to do some of the manipulations that allow us to study other organisms, their genes and their genomes. For example, the Human Genome Project could not be completed without the help of bacterial genetics and bacterial cloning that we used to make copies of the human genome, so we're able to do sequencing and things of that nature. In addition, the knowledge we gain from studying bacterial genetics and bacterial genomes is widely applicable to other organisms as well. So before we get too far into the weeds, I want to do a little housekeeping and clarify a couple terms that we're going to use. Prokaryotes, that's going to be referring to bacteria and cyanobacteria, which is just a subtype of bacteria. They have DNA that's not bound by a membrane. There's no nucleus in prokaryotes. They have single molecule, double-stranded circular chromosomes. So they're very small compared to eukaryotes, which is animals outside of bacteria. And they reproduce asexually. So they just divide themselves, right? We'll talk about kind of a little form of gene transfer that's similar to sexual reproduction in this chapter, but generally they reproduce asexually. Viruses parasite bacteria. So they, besides parasitizing, I should say, humans, they also do this to bacteria, where bacteriophages, which are viruses that are specifically aimed at bacteria, what they do is they inject their DNA into the bacteria, and that's how they reproduce. And so we'll see a little more about that, but if I refer to phages or bacteriophages or viruses, those are all synonyms for the same thing in this chapter. So there are 4 types of ways that bacteria exchange DNA. Whether that's uptaking DNA or donating DNA, etc. We'll talk about all 4 of these in this chapter. The first one I want to mention is this transformation at the top here. This one was vital, very vital for the Human Genome Project and any sort of DNA work that you might do in the lab. Cloning is all because of transformation. All the processes of making large copies of DNA by harnessing bacteria, that all relies on this transformation process. And that's when bacteria cells will just naturally uptake DNA that they find in their surroundings. There's ways we can expedite this process. Electroporation is a way that we put kind of voltage through the bacteria, and it forces them to take up this DNA quicker. There's also calcium and other ion-derived ways of doing it. We'll talk about that a little bit more in the actual class and discussion. The second one I want to hit on is transduction, and that's, like I said in the last slide, this viral way of introducing DNA. Another thing that we can harness is using these viruses to package genes of interest for us and be the vector to deliver this DNA into a bacterial cell, but they'll do it naturally too. That's their means of reproduction, which we'll also get into this chapter. And then there's 2 forms of conjugation here. The first is when 2 bacterial cells will join up with this pili, which we'll talk about in a little bit. And one is that they'll donate a plasmid. A plasmid is a small circular piece of DNA that is not the main genome. It's kind of an accessory piece of DNA that is found in bacteria. The other way is there can actually be genome transferred during conjugation too, and this allows for crossing over and things that we talked about in those previous chapters to occur. And so they're both use the same mechanism, but one transfers a plasmid and the other transfers the chromosomal DNA of this these bacteria. A lot of you may have taken microbiology at this point, but not all of you. So we're going to do a little bit more housekeeping and talk a little bit about bacterial colonies and how they work. And so to derive single cell bacterial colonies, so these are colonies or giant populations of bacteria that are all derived from one single cell. And thus they should have nearly identical genomes. You might have a mutation here and there that switches things up a little bit, but generally they should all be clones of each other, because remember, bacteria for the most part reproduce asexually. So to do this, we start with a suspension of bacteria cells. So we have a test tube full of this liquid here, and there is just a bunch of bacteria cells floating in. What we do is we take a small amount, like a little drop, of those bacterial suspensions, very dilute, so there's only a handful of bacteria in that drop that we add, and we spread it all around this petri dish here, right? And so all those little bacteria, they should all be spread singly. I'm making little dots on here to represent my spread out bacteria. They should all be spread out individually across this plate. So here they are pointing to my little bacterial colonies again. And so you should have single cells where it's pointing to that. And then you incubate them for a day or two. Usually if you use something for cloning like E. coli, you just do it overnight and you'll be able to see them the next day. Some other bacteria take a little bit longer because their doubling time is a lot less, or a lot more, I should say. So it takes a lot longer for them to double and for those colonies to appear. So 1 to 2 days we incubate them. And we should have visible colonies where each one of these colonies here, we got 1, 2, 3, these are all colonies. Each one of those is derived from a single bacteria, so they're all identical to one another. And so here's kind of the overview of that whole process. And this is important because if we are looking at cloning a gene or making libraries where we put different individual pieces of DNA into all these bacteria to cover an entire genome, we need, first of all, enough of these bacteria where we can do DNA extractions and actually have molecular material to work with. And we also want them all to be derived from one cell, because if we're looking at a gene where we are cloning a gene, we want to have a large number of bacteria that all have that gene in it. If they don't have that gene in it, that's going to be a problem. It's just wasting our time and diluting our samples. So one thing that we can do to ensure that these bacteria grow with our genes in them is to use these molecular selection markers. And what they are genes that the bacteria naturally have. And if they are positive or minus, that means they are requiring certain media to grow, right? So if you are lac minus, in this example down here, it can't utilize lactose as a carbon source. So bacteria that are lac minus, if you put them on a media that only has lactose as their source of carbon, they're not going to grow, right? In addition, there's a lot of antibiotic selective markers. So down here we have streptomycin, and so we have resistant and sensitive. So if we have a bacteria, say we want to put a gene in a bacteria and clone it. If we put that gene into the bacteria, in addition to this streptomycin gene, so we have them kind of like back to back, then anything that grows on a medium that has streptomycin, then we know that it got our gene as well, because we put both those in together. So these selective markers are very important for being able to grow and select different bacteria based on what we're looking for. In addition, there are dyes that are specific to some of these genes. One that we'll talk about is XGAL, which is a beta-galactosidase gene. Here's an example of a plate where we have lac plus and lac minus cells. So lac plus being all these purple lawns or colonies over here. Lac minus being these individuals that were not able to create that pink or purplish color. So we also have visual cues beyond survivability that tell us what genes are inside these bacteria. So as I stated earlier in the lecture, this question is a little faux pas because of that. Do bacteria have a process similar to sexual reproduction? As I mentioned earlier, there is a process that is similar, but we're going to kind of go through a thought experiment here. And so we have two strains of bacteria. Strain A on the top here is negative for MET and bio, two metabolism genes that are important for reproduction. And strain B minus is positive for MET and bio, but negative for THR, LEU, and THI, which are amino acid genes. And so in order for a bacteria that is minus for one of these genes to reproduce, MET or bio or leucine or any of these amino acids that are missing, those need to be supplemented to the bacteria because they're not able to biologically process them themselves, right? So neither, unless strain A or strain B are supplemented with some of these things that they're minus for, they're not going to grow on just regular media. So if bacteria are able to perform some sort of mixing of genes between individuals, kind of akin to a sexual mating, then we would expect that a mixture of the A- strain and the B- strain would result in some progeny that are wild type, meaning that they are plus for all 5 of those genes. So in our first part of the experiment here, we would do our controls, right? So we would start by taking our A strain here and growing a broth of it. So we have a whole bunch of bacteria, and then plating it. And if that's plated on minimal media, meaning we don't supplement MET and bio to aid in the growth of these bacteria, we should end up with no colonies, right? And so if we do the same experiment with strain B over here, we grow them into a broth media. So we grow a whole bunch of these cells. We wash them, and then we plate them again on this minimal media. So we're not supplementing THR, LEU, or THI. We end up with no colonies once again. However, if we mix A and B, A over here and B over here, into a broth that contains both of them, if there was some sort of exchanging of genes between these two strains of bacteria, then when we would wash the cells and we plate them. Once we plate them on minimal media again, we see these colonies form. So these big circles are showing that there are a whole bunch of colonies forming. And that means that these colonies had to be from bacteria that are positive for all 5 of those genes that are needed for this phototrophic growth. So theoretically, that would mean that there's some sort of exchanging of genes. And this is actually what we do see. So we do know from these early experiments of mixing 2 different genotypes of bacteria that they are able to exchange genes. So now that we know that the bacteria do exchange genes, now we need to know how it happens, right? And so one simple thought was that, okay, maybe these bacteria are expressing their genes as RNA and just letting them out into the liquid media, and then the neighboring bacteria just absorb those genes. Because we saw early in that transfection idea that bacteria can just take up DNA and RNA from the surrounding environment. We can expedite that with things like electroporation by putting currents through them. But what if the bacteria are just making copies of all those plus genes and then putting it into the media, and then they're taking it up with one another? So there's this simple experiment done where they took this U-shaped Eppendorf tube, or test tube, I should say, and put strain A in one side of it, strain B in the other side of it, and put a very fine filter in between. So this filter had holes in it that were small enough to let DNA and little pieces of RNA flow through it, but not small or not big enough to allow entire bacteria. So all the bacteria from strain B over on this side, they stay floating over on this side. All of the bacteria from strain A are on the left-hand side here, floating around, but they're not allowed to intermingle. All the little proteins and pieces of DNA and RNA and little polymers like that are able to be exchanged between, but not the entire bacteria. So what that U-shaped tube experiment indicated was that no bacteria grew when they were separated by that filter, meaning that those bacteria, in order to exchange genes to become fully able to grow on minimal media, they had to actually come in contact with one another. And so once we got to the stage of being able to use microscopes, we were able to see that these little structures form in between different bacteria. And these structures are called pili, P-I-L-I, and they're little bridges that form between two bacteria. And so if you have that filter in between, these pili cannot go through that filter, so you actually have to have two bacteria cells that are able to come within physical contact of one another to exchange this genetic material. So the molecular machinery or method for this is those pili, and this is created through the F plasmid. And so the F plasmid is a little non-chromosomal piece of DNA that it's a little circular piece, so it's a plasmid. It floats around inside this bacteria, and that makes that bacteria F positive, F plus. So it means plus meaning it does have that plasmid. And what that allows is this bacteria here to form this pili, or these pili, or pilus for singular, and it will interact with an F negative bacteria. So F positive only interact with F negative bacteria. So it's kind of like almost, you can think of it akin to reproduction in any sort of mammal where you need a male and a female, where you have two opposites. And you can't have two female lions make a baby together, or two male lions make a baby together. You have to have a female and a male. So you have to have an F plus and an F minus bacteria. The F plus will create this pilus, and it will attach the F minus cell. But what's interesting compared to the sexual reproduction in mammals is that what happens is this F plasmid will replicate itself through this pilus. And so you'll have a copy that stays inside that first bacteria, and then you'll have a new copy that's made inside the second one. So this will eventually become a full round plasmid over here. The reason that is interesting and different than sexual reproduction is when we get this plasmid that is formed on the recipient, formerly F minus cell over here, this second cell now is F plus. So we end up with originally an F minus and an F plus, and at the end of this transfer, we end up with two F plus cells. On rare occasions, when this F plus plasmid, or this F plasmid, is passed on to a F minus cell, sometimes that plasmid will then be integrated into the actual chromosome. So in this first example, this F plus up here, we have it, remember, it's on a separate plasmid, so it's just sitting there kind of by itself, floating around. The genes still get transcribed because there's no nucleus to keep it separate from the chromosomal DNA. But once in a while, after this transfer, that actual F plasmid will get incorporated into the host chromosome. And then we get which is called something called an HFR strain of bacteria. HFR stands for high frequency of recombination. And so what this means is that these cells can create up to as many 1,000 times as many recombinants as the normal F plus bacteria. So remember, recombinant DNA is where we exchange different genes from one chromosome to another. And we'll get into the process of how that's done in bacteria here shortly, but just remember HFR means these things recombinate like crazy. So how does this crossing over occur in bacteria? So remember this HFR strain. So that was that strain of bacteria that has the F plasmid that was integrated into the host chromosome. So these integrated F plasmids are still able to form this pilus that we had talked about that allows interaction with an F- strain. What's different is that in these HFR strains, that plasma that usually, the F plasma that usually crosses over that pilus is actually on the host chromosome of this HFR bacteria. Now, as that F plasmid crosses that pilus into the F- bacteria, it also is dragging along all the rest of this chromosome with it as well. And so it will bring a large chunk of that chromosome into that recipient F- bacteria. Then this piece of chromosomal DNA that is crossed over from that HFR bacteria will be allowed to cross over with the host chromosome, or the full chromosome of that recipient F- bacteria. And so similar to eukaryotes that we saw in the last chapter, crossing over can then occur in the same way where you exchange gene for gene between these two. But what's interesting with bacteria is that in order for a piece of DNA to remain in bacteria, it has to be circular. So in this chunk of chromosomal DNA that is passed over to the F- bacteria, that chunk is then lost, is degraded by the host cell, and only the genes that were recombined are what's left in that recipient cell. So in this crossing over process, we have to remember too that it's not instantaneous, that all that HFR, the F plasmid portion of it, just flashes across that pilus into the recipient cell. It actually occurs in a process. And so we can actually use this process of HFR chromosomes being passed from a HFR strain of bacteria to an F- strain. We can use this process to determine the actual order of genes on the chromosome. And what's cool about this experiment is that it used just a household kitchen blender to do this. And so what happened is they would take two strains of bacteria, one HFR strain and one recipient strain, and at certain time points, they would break that conjugation, those pili that are between the HFR and the F-, they would break them apart by using a blender to just mechanically shear the bridge between these two cells. And when they did that, they could look at selective markers, like we talked about before, growing on antibiotics or minimal media, etc., and see what grew. So they saw that after about 10 minutes, if they broke apart those bridges, that almost 100% of the X conjugates, so both the donor and the recipient cells, had this AZI resistance. After about 11 minutes, they saw that about 80% had resistance to this TON. And then at about, we'll say 18 minutes, that 40% had the ability to grow on lactose, which was not present before then, right? So, and then at about 23, we'll say, minutes, they had the ability to grow on galactose. Now you'll notice that as we go further down the chromosome, nothing appears to get as high efficiency or frequency as the ones that crossed first. So galactose and this AZI resistance are not on the same level. They kind of both plateau, but one plateaus at about 90%, the other plateaus at about 42%. And the reason this is because even if we were allowing these conjugations to occur fully, there's unknown forces that cause these conjugations to break naturally. So they won't be conjugated forever, even if we gave them the perfect environment. Eventually these conjugations will break, and you never have an entire HFR chromosome passed to an H minus or a F minus. It always dissociates before that. But using this information here, we can see that the order of the chromosome, or genes on the chromosome, you'd have the F factor that caused that initiation and that formation of that pili, and then you have AZI, TON, LAC, and GAL proceeding further away from that F factor. So here we have kind of a characterization of what was happening in that last graph. So as I said, the F factor, or the origin, as we call it, on that F factor, is what proceeds across this formed pilus first. And then when we looked at that last graph, if we disrupted those physical connections at about 11 minutes, we would cut the bridge right here. And we'd have only that AZI, which was purple, and then TON, which is blue, will have made it across in that time. The other genes, the galactose genes, etc., didn't have time to proceed across. However, if we waited for 17 minutes, then broke the conjugation, then we had the first 3 genes proceed across the bridge, but galactose, again, did not make it. And then lastly, if we waited at least 25 minutes, then all 4 of the genes we were looking at proceeded across that bridge. And so from this, we can denote, obviously, the order of genes is the purple, blue, orange, and then green that correspond with the time frame in the last image. So another interesting observation that was made about these HFR strains was that some of them appeared to have genes in different order. And so here we have the origin, which is O, and then the rest of the F factor. And then we had all those genes that we talked about, THR, PRO, LAC, etc. But they all are mixed up here. So we have THR, then PRO in the first one, but in the second one we have THR, then THI. In the second one, or the third one, 2', we have PRO, then THR, which is backwards of this initial strain. So we have them all mashed around, and there wasn't a good reason to understand how these genes are shuffling order like that. So what we came to find out is that the reason this is because those F factors will insert at a specific locus, and it doesn't matter what they can insert forward or reverse, so their origin or where that they're facing can change depending on the insertion event, and they can really insert at a whole bunch of different locations throughout the host chromosome's positioning. And so thus, depending on where you insert will determine what order the genes are that are being transferred. So if we go look at those original 5 sequences that were on the 2 slides ago that all appeared in different order, well, they can all be explained by changing either the orientation, left to right or right to left, orientation of the insertion of the F factor, or between what genes those the F factor inserted itself. So in all 5 of those cases, which we have again drawn at the bottom here, H, 2', 1', 3', and 3, 1, 2, those 5 orientations all match up to the same circular chromosome. So all 5 of these circular chromosomes have genes in the same position, but if you read in the order of the arrow of this F factor, depending on where you inserted, it appears that you have a different order. So the order on the chromosome is the same, but the order of transfer, when those HFR transfer their host chromosome to an F- strain, that all depends on the integration site of that F factor that made it an HFR strain. So just to recap conjugation here, remember we looked at the second or third slide that we're talking about the 4 different types of DNA transfer. 2 of them were conjugation. So to recap, we can have chromosome transfer, which is where that F factor was integrated into the host chromosome of that bacteria to make it an HFR cell, which in this case, or HFR standing for high frequency of recombination. That HFR will then conjugate with an F minus individual, and it will transfer not only the F factor, but also some of the chromosomal DNA. And then depending on how much of that DNA is transferred and if recombination occurs or not, because it doesn't always have to happen. Sometimes that chromosomal DNA can be transferred over and recombination does not occur, and thus it just gets degraded, and that F minus cell stays that same F minus cell. So the outcome is we can either have an F minus cell that has experienced recombination, or we can have one that has had no recombination. The second type of conjugation that we talked about was plasmid transfer, and that is when that F factor is not integrated into the host chromosome. So we have an F plus, not an HFR, an F plus, so it has that factor, but it's separately on the plasmid. And what that does is during conjugation, that F factor transfers itself across to that F minus, making that F minus an F plus as well, but none of the chromosomal DNA is exchanged. Then, once that F factor is transported across, it can either be integrated into that recipient cell to make that recipient cell an HFR, or the plasmid stays separate and is just an F plus cell itself now. So we have the donor as an F plus and the recipient are both F pluses at that stage. One thing to note when talking about prokaryotes and bacteria versus eukaryotes is with recombination, you need to have double crossovers occur for there to actually be a viable product. If you have a single crossover, which can happen in mammals and eukaryotes, etc., if you have a single crossover, what happens is you end up with a linear piece of DNA. Remember, that is not viable in bacteria. All pieces of DNA need to be circular in bacteria for it to be viable. And so if you have a single crossover event, you produce single-stranded non-circular DNA, and thus that DNA will be degraded and that cell will die. So you have to have double crossovers in bacteria to be viable. So previously we talked about recombination mapping, but this also happens in bacteria. And so we're able to look at the distance between genes and bacteria through recombination mapping, just like we are able to in eukaryotes. And so in this case here, we have 3 genes, leucine, arginine, and methionine, that are transferred over to a host chromosome that is negative for all of these. So the F minus recipient was negative for leucine, arginine, and methionine. So now if we were to look at the percent of offspring that arise from this conjugation event, we see that 87% of offspring are positive for all 3 genes, 9% are positive for leucine and arginine, and then 4% are positive for just leucine. So from this information, we can deduce what the order is of the genes and what type of crossover events occurred because of the distance between these genes. So I've poorly drawn the percents that we saw on the last slide onto this side. So the leucine positive and then arginine and methionine negative example, we saw 4% of those individuals. And so the reason we see 4% is because we have the origin that proceeds across first, and then the first gene across would be MET, then arginine, and then leucine. So the reason this is only 4% is because the conjugation had to happen a long time, which, remember, they can break at any moment, and so genes closest to the origin get transferred at a higher percentage. So this means that it had to happen a long time to ensure that leucine was across the bridge before the bridge broke. And then the crossover event had to happen between arginine and then after leucine for only that gene to be transferred. So this is a very rare occurrence, right? So now, in addition, the distance between leucine and arginine is fairly small in this case, that very few recombination events occur in between them. So now we look at the second example where we have leucine and arginine get recombine at 9% rate. And so again, this requires that all 3 genes make it across, and only leucine and arginine get crossed over, and not methionine, which would be the highest amount. So for this event to happen, you're counting on recombining of very few successful transfers, because this leucine, remember, is transferred at a less successful rate than methionine just because it's further down the chain. And so you see that it's 9%. And then the most common thing to happen is, because this region between methionine and the origin, and then after leucine all the way down to however far the longest chain is, those are the biggest regions that allow for crossing over to happen. So for all 3 of these to cross over, happens at the largest event, or the largest possible outcome, and that's at 87%. Now, there was one combination that occurs that was not even shown on our graph, and that's for leucine to be recombined and methionine, but not the middle gene, arginine. And so what this requires is for 2 crossover events where only leucine gets transferred and only methionine gets transferred, but not arginine in the middle. And so for this double crossover of genes, remember, we need 4 recombination events to make sure that it's linear. So this occurred in absolutely 0 of our outcomes, or our samples. And so if you increased the sample size tremendously, it's possible that you would see this, but it's a very, very unlikely event. An additional note on that last slide, if it is confusing, we will look at this in class too and discuss it. It is a little bit difficult to explain in a lecture format without people asking questions, so don't fret if you don't understand it. We'll talk about this in the actual lecture. So just as F plasmids can integrate themselves into a host chromosome, they can also outloop themselves. So they can also kind of extract themselves from these host chromosomes. And sometimes, depending on the homology of the sequences that are in the F plasmid and in the host chromosome, we get what's called faulty outlooping. And what that means is that for outlooping to occur, there has to be 2 very similar homologous pieces of DNA that allow for this matching up and kind of crossing over to occur. So here, it's kind of hard to see. The colors red and orange are very close, so it's hard to see. But we have an integrated F factor, and we have these 2 homologous sequences here. And so the outlooping should naturally occur where those 2 kind of pinch off and allow this chromosome to come back together, as in this first picture here, and the F factor just be released. Well, if you notice on this B example here, we also have another piece of homologous DNA. It's the same as those other 2 orange factors further down the line. So if during this excision, this number 1 over on the left here and number 3 over on the right pair up instead of number 1 and 2, all of a sudden we've incorporated this lac gene onto the F plasmid. And so that's an example of faulty outlooping, where all of a sudden these F plasmids can pick up pieces of chromosomal DNA and carry them with them. And so that F plasmid will then go and do its normal F plasmid thing of finding a F minus strain and passing that F plasmid from one to the other. But it can also take some of these chromosomal genes, in this case an antibiotic resistance gene, with it. So there's quite a few examples of these plasmids that have incorporated functional and sometimes dangerous chromosomal genes into those plasmids. What becomes a very dangerous bacteria is when these plasmids start to accrue antibiotic resistance genes or other genes like enterotoxin production down here, where they're able to almost kind of like a virus, where a virus passes from people to people. This resistance to antibiotics can be passed from bacteria to bacteria, and you start to gain this community resistance to antibiotics, and it can be kind of dangerous. This also is a big thing in plants, where this T1 plasmid, which is the formation of tumors in plants, that has also been integrated onto a plasmid. Luckily, scientists have found a way to actually use that to their advantage, and this is actually used for making GMOs and things of that nature, gene transfer, but it can also be very dangerous to ecosystems. So another type of plasmid, similar to an F plasmid, this is this thing called an R plasmid or an R factor. These R factors are plasmids that through this outlooping process has accrued multiple antibiotic resistance genes, sometimes up to 8 antibiotic resistance, and it can be passed from bacteria to bacteria. So this was discovered back in the 50s by Japanese scientists in Shigella, and they found that these bacteria were able to accrue antibiotic resistance and pass them sometimes to other species of bacteria, this resistance. And so this is a very kind of concerning thing. You hear about these superbacteria and these antibiotic resistance, MRSA, and things of that nature. So it's something that's very concerning, is their ability to accrue these different antibiotics. And so that's one reason that you have to make sure to take your antibiotics the full course that the doctor tells you, and make sure that you're not using them less and selecting for more of these antibiotic resistant genes to be accrued into F factors. Moving on from conjugation, another way that DNA can be taken up into a cell is through this transformation process. As I said earlier, this is where DNA from surrounding solution or agar media or wherever it may be is exposed to a bacteria, and through these holes in the cytoplasmic membrane of the cell, the DNA will pass through in a single strand nature. There's these DNA degrading enzymes that will degrade half of the 2 strands of the DNA, and that single strand is then integrated into the cell, and either or that cell then will insert that DNA into the host chromosome, and then the replication machinery will come in and fill in the second of those 2 strands. And so this is a very rare process. However, this is how we clone genes and things of that nature. And so when we want to put genes into bacteria and have them make a whole bunch of copies for us, we put that DNA in a solution with the bacteria. And we found ways to make this process of passing that DNA from the outer cellular matrix, the solution, into the cell much more frequently than this process happens naturally. And that's how, like I said, there's a freeze-thaw method where you get bacteria super cold and then warm them, and then super cold and warm them over and over. And that causes these little holes in the membrane to occur, and the DNA will pass through. Or through electroporation, which uses electrical current to create those holes in this plasma membrane. But this is a very, very important part of molecular biology if you're going to do any work with DNA, this idea that bacteria will take up pieces of DNA from their surrounding environment. So we've covered 3 of the 4 methods of DNA transfer between bacteria: conjugation with the plasmid, conjugation with a chromosome, which that F plasmid was integrated into, and then transformation. But the last thing we're going to talk about are other vectors that can carry DNA between bacteria. And the way that that DNA is transferred is through phages, as I alluded to in the introduction. Bacteriophages, phages, viruses, all different sides of the same coin here. So this is a picture of a phage. We have some different components. The head is where the DNA is stored. There's a neck and collar that's attached to this core, and around the core is this sheath of molecules. There's kind of this end plate at the bottom, and then these fibers that are like legs that are used to attach to the bacteria. Here's an electron microscope picture of these phages. You can actually see the heads, the sheath surrounding the main component there, and then also those spindly legs that are coming off the bottom of the bacteria. So this is an actual photo of a phage infection, and so we can see the heads again of the bacteria, the sheath here, and if you look very closely, you'll see that there's actually something protruding into this cell that is being attacked by these phages. And this is actually the DNA that is being passed from the head. So it starts up here. This is my squiggly red DNA. And when that phage locks onto that bacterial cell, it'll pass through and into the target of this phage's attack. Throughout our use of this textbook, there'll be times that I point out some things that are very odd or figures that were not caught by the editors very well of this textbook. And for some reason in this figure they have this chromosomal DNA that is trying to escape the bacterial cell through who knows what. Just imagine that this is fully inside the cell, this circular piece of host DNA, or of bacterial DNA. I'm not sure what the publishers were aiming for here, but we have an uninfected cell with by no means the DNA trying to escape. And now we have this free phage floating around looking for a target or something to inject its DNA to. So it finds this bacteria and it enters the lytic cycle where it's attached to the outside of the bacteria. And it's a little difficult to see in this figure because it's very small, but you can see right here, this little blue line, that's the phage actually starting to inject its DNA into that bacteria. Now we have this DNA that has been injected along with some other proteins, and that causes the bacteria's host chromosome to be degraded, and this phage DNA to be in there, where the replication and transcription and translation machinery of this DNA or of this bacterial cell will start to form proteins for replicating that phage. So you can see there's a bunch of phage proteins, like little pieces of head and little sheaths and little legs, all floating around in this infected bacteria cell. Now that all these parts are being made, the phage will kind of hijack the rest of the machinery of the cell, and will start to assemble these phages, and will also incorporate more DNA. So it's used this DNA replication machinery from this host cell as well, and so it's made a whole bunch of copies of itself within this host DNA or host bacteria. Lastly, the phages will reach this critical point where they've made so many of them within the cell that it will lead to this lysis of the cell, where all those phages are then free to float around in the media again like our original phage was and look for more host targets. And so this is what leads to viral infection, where you have this uncontrolled breakout of these phages being produced and then infecting more host targets. So we can see this with our naked eye if we have a plate of bacteria. So this is what we call a lawn of bacteria, where the entire petri dish is just wall-to-wall pieces of bacteria. But we've introduced phages into that, and so these little dots are actually places where there's no lawn of bacteria, and that's because it's a scorched earth place where the bacteria has been killed by all the viruses And so hundreds and hundreds, maybe thousands of bacteria in this little dot have been killed by this, this transferring of the virus. So now we've talked about how viruses work, but how does this transfer bacterial DNA between two individuals? And so what sometimes can happen is this process begins where we have a virus that is transferring its DNA into a bacteria. And again, we have this, the host chromosome here is broken into parts. And so we have this A+ gene floating around and this B+ gene that were once part of this circular chromosome. And the virus has started to replicate, pieces of its head and its sheath, and the legs are floating around there. And when these viruses are reassembled, sometimes just by random chance, the head will incorporate actual chromosomal DNA from that bacteria that was infected. So for the most part, most of these, we have this red DNA, which was that virus DNA that was replicated. But in two instances here, we have the actual host chromosome DNA that was incorporated into the virus. Now this virus proceeds on just like normal because all the machinery of the virus is the same, except for it just has carrying a different package. And so this virus that had that A+ gene will go infect a piece of bacteria that the recipient piece of bacteria it's infecting has a copy of the A- gene. And that allows for an A+ and an A- to be in the same cell, and allows for the recombination of the A+ to take the place of the A- gene in that recipient bacteria, causing the recombination of the genes in that bacteria. So that wraps it up for our chapter 5, talks about bacterial genetics, and we'll proceed with chapter 6.